Imagine you spend like 10 years of your life and $2 billion meticulously forging this perfect key to unlock a highly specific vault.
Right.
You finally finish, you walk up to the building, and you realize you've been trying to unlock a painted-on door. Like, there is no keyhole.
Yeah, which is just, I mean, it's a brutal scenario.
It is. But that brutal scenario is the reality of the modern pharmaceutical industry right now. Even with all our cutting-edge technology, 90% of the therapeutic candidates that enter clinical trials just fail. 90%.
It's staggering.
Right. And the primary reason for that failure rate isn't that the chemistry is bad. It's that we fundamentally fail at finding the right biological targets to begin with.
Exactly. It's a massive attrition rate. And it really highlights this critical flaw in how we've approached drug discovery for the last century.
Yeah.
We've basically been relying on this process of highly educated trial and error. But what we are looking at today for this deep dive is a total paradigm shift.
A completely new way of doing things.
Right. We are moving into an era of what is broadly termed scientific intelligence. And specifically, we're going to unpack this framework called Pharmaceutical Superintelligence, or PSI.
PSI. Okay, so what exactly is that?
Well, it's this orchestrated suite of AI agents working together to solve one of the absolute hardest puzzles in biology, which is finding and then targeting what are known as cryptic pockets.
And for you listening, to really grasp why this is so revolutionary, we have to look at what a cryptic pocket actually is, right? Because traditionally, to design a drug to stop a disease, let's say it's a malfunctioning protein causing cancer, you need a place on that protein to plug the drug in.
Right, a binding site.
Exactly, a binding site.
Yeah.
But what happens if that plug socket is completely hidden? Like, cryptic pockets are these structural vulnerabilities on a disease-causing protein that are completely invisible when the protein is just, floating around in its normal resting state.
Which is most of the time.
Right.
Yeah.
They only pop open and become detectable when a drug actually binds to them.
Which creates this intense chicken and egg paradox for researchers. I mean, how do you design a custom molecule for a pocket that you literally cannot observe until the molecule is already inside it?
You really can't.
No, you can't. For decades, discovering these sites was basically a lucky accident. You couldn't just look at a static 3D image of a protein and see the pocket because the protein looks like a solid, impenetrable boulder.
Right.
So to unlock that hidden door, you first have to figure out where the invisible seams are.
And that takes us to the very first layer of this multi-agent AI suite. Right. This system built to just scan the entire landscape. It's a proteome-wide radar called All-Else-Sites.
Yes.
We're talking about scanning the entire human body's catalog of proteins here.
All 20,420 of them, roughly speaking.
That's wild.
It is. All-Else-Sites is the suite's pre-screening layer. And what makes it so remarkable is its throughput and really its universal applicability. It is a unified sequence-based framework.
Meaning it works on basically anything.
Essentially, yes. It's capable of identifying potential binding sites across all drug modalities. So whether you're trying to design a traditional small molecule drug, a peptide, or even working with DNA and RNA, All-Else-Sites can find the target.
Okay.
And here's the kicker. It screens the entire human proteome in just 16 hours.
16 hours?
Running on a single GPU.
Okay, let's unpack this for a second, because that sounds like magic. I mean, there has to be some deep logic here. A protein sequence is essentially just a one-dimensional string of amino acid letters.
Right, correct.
It's like a long sentence in a book. So how is this AI taking a flat line of text and predicting a hidden three-dimensional pocket without, running the heavy 3D math to fold that protein up?
Well, it accomplishes that through the sheer brilliance of its neural architecture. All-Else combines a gated convolutional neural network.
A gated CNN.
Exactly, gated CNN with the transformer architecture.
Yeah.
And, if you think about how large language models read text, they don't just look at one word at a time, right?
Right. They look at the context.
Exactly. They look at the context of the entire paragraph. The transformer in All-Else does the exact same thing, but with amino acids. It captures the global sequence features. It understands that an amino acid at position 10 might heavily interact with an amino acid at position 400 once the protein naturally folds.
Oh, I see. So the gated CNN acts like this hyperfocus scanner looking at the immediate neighborhood of amino acids, while the transformer is kind of zooming out to see the long-range relationships across the entire sequence.
You've got it. The system is essentially reading the grammatical rules of the protein.
That's a great way to put it.
It recognizes the sequential signatures and local contextual patterns that historically tend to form pockets under stress, so it completely bridges the gap between sequence-based and structure-based methods.
So it doesn't even need the 3D physics.
Right, it doesn't need to spend days calculating how the protein bends and twists in water because it has already learned the abstract blueprint of what a hidden structural weakness looks like purely from the sequence code.
Okay, so Allsites has given us this massive proteome-wide list of potential targets in just 16 hours.
Right.
We now have thousands of these flag sites. But this introduces a huge logistical bottleneck, doesn't it?
It absolutely does.
Because if we want to verify these pockets using actual 3D physical simulations, we can't run them all. Simulating just a microsecond of protein movement at the atomic level takes an immense amount of supercomputer time.
Oh, yeah. We would literally melt our servers trying to simulate 20,000 proteins.
Right. So how does the system physically triage what to simulate next?
To solve that compute optimization problem, the suite brings in a specialized algorithm called Pocket Miner.
Pocket Miner.
Yeah. And Pocket Miner performs what we call residue-level criticity prioritization.
Okay, let's break that down. A residue is just a single amino acid building block within that larger protein chain, right?
Correct. So all sites highlighted the general regions, but Pocket Miner scores the exact mathematical likelihood of any specific residue being the physical hinge of a cryptic pocket.
I look at this almost like structural engineering. Like all Allsites looks at the blueprint of a massive sustension bridge and highlights a general area that might be structurally weak. But Pocket Miner is the algorithm calculating the exact tension on every single rivet. It tells the system, look at rivet number 402. If pressure is applied there, the steel will buckle.
That is a really highly accurate way to visualize it. What's fascinating here is that Pocket Miner is looking for the structural weak points to prioritize the workflow.
Right.
By identifying those high-probability residues first, the engine directs its heavy computational artillery, which is the molecular dynamics or MD analysis, only to the protein regions that actually matter.
Because why simulate the whole bridge when you only need to simulate the broken rivet?
Exactly. You are saving immense computational time and cost by not running deep physics simulations on parts of the protein that are essentially solid brick walls.
Which leads us to the next massive hurdle. We have the exact stress points prioritized by Pocket Miner. But predicting a weak point on a 1D sequence is very different from proving it exists in the 3D physical world.
Very different.
The system now has to physically force this virtual door open and map out exactly how it moves. We're moving out of sequence prediction and into heavy physics-driven sampling.
Yes, and the AI suite has an entire arsenal for this. It starts with a tool called AF2-Rave.
AF2-Rave, got it.
Yeah. This is where the predictive layer gets augmented by actual structural data. It leverages AlphaFold, the famous protein folding AI, but it uses it in a very specific way.
How so?
Well, instead of just asking AlphaFold for one static resting picture of the protein, it generates an entire ensemble of target structures.
Like a flipbook of the protein breathing and shifting, capturing it in different poses where a pocket might be partially yawning open.
Yes, an ensemble of slight variations. Then, AF2RV takes that flipbook and iterates between molecular dynamic sampling, which simulates the physical movements over time, and autoencoder-based analysis.
Autoencoders being neural networks that compress data to find patterns.
Right. So by bouncing between the raw physical simulation and the pattern-recognizing AI, the system gradually builds what biophysicists call a collective variable, or a CV.
Okay, let's break down the mechanics of a collective variable for the listener, because it's a pretty dense term. This is essentially a mathematical progress coordinate, right? It maps out the actual choreography of the pocket opening.
Think of the collective variable as the exact sequence of mechanical steps required to open a complex lock.
Okay.
The protein doesn't just spontaneously pop open all at once. First, a surface loop of amino acids has to swing out of the way, which then allows a deeper helix structure to slide to the left, which finally exposes the cavity.
Right.
The CV mathematically describes those key underlying physical motions.
But sometimes we aren't dealing with a simple loop swinging out of the way, right? I mean, sometimes the protein requires a massive structural shift to expose a pocket. We have whole helixes that need to be displaced.
Oh, absolutely.
And the sources point out that for those heavy-duty jobs, the AI uses a method called SWISH.
Yes, SWISH. It stands for Sampling Water Interfaces Through Scaled Hamiltonians.
That is quite the acronym.
It is. But the concept is brilliant. When a cryptic pocket is buried deep inside a protein, the tight atomic packing often prevents normal simulation techniques from ever finding it. The forces holding the protein together are just too strong.
So what does swish do?
It temporarily alters the physics engine of the simulation. It artificially scales the interactions between the protein and the surrounding water.
Oh, wow. It's like taking a sealed cave system and artificially increasing the water pressure to 1,000,000 psi just to see where the water forces a microscopic crack to widen.
That's exactly the right concept.
Yeah.
By tweaking how aggressively the water interacts with the protein surface, swish forces the water molecules to act like wedges.
Driving into the protein.
Right, driving into and exposing buried cavities that organic probes would completely miss under normal physical rules.
That's incredible.
And once the cavity is forced open, the system needs to verify it. And that brings us to MSMD.
Mixed solvent molecular dynamics.
You got it.
This is hands down my favorite part, like the most fascinating part of the physics layer.
Mm-hmm.
So we forced the pocket open with swish. Now, MSMD takes the simulation and floods it with low concentrations of organic computational probes.
Yes.
Essentially, it pumps the virtual water full of small, drug-like fragments, like virtual benzene molecules. It reminds me of automotive engineers spraying colored smoke into a wind tunnel to reveal the invisible aerodynamic currents over a car's chassis.
The wind tunnel analogy captures the diagnostic nature of it perfectly. Because as the simulation runs, the AI tracks every single molecule of that virtual benzene. It is specifically looking for areas of high probe occupancy and long residence time.
Let's focus on that phrase, long residence time. Why does it matter how long the benzene stays there? I mean, if it touches the pocket, isn't that enough?
Not at all. Proteins are dynamic, you know.
Mm-hmm.
They are constantly vibrating and forming tiny momentary divots on their surface. If a benzene molecule drifts into a shallow divot and immediately bounces back out into the water, that divot is completely useless for a drug.
Because the drug wouldn't stick.
Right. A real drug needs a stable anchor point. If the benzene gets wedged into a crevice and stays there for a long time, that long residence time, the AI mathematically proves that this isn't just a random fluctuation.
It's a real target.
Exactly. It is a stable, actionable, hidden pocket.
So at this point, the AI suite has run a proteome-wide scan, triaged the weak points, mathematically mapped the opening choreography, and physically proved the pocket can hold a drug-like fragment.
Yes.
But here's the ultimate question for you to consider as you're listening. Just because a dent exists on a protein, does that mean plugging a drug into it will actually cure the disease?
And that is the multi-billion-dollar hurdle that historically traps pharmaceutical companies.
Right.
You can design a brilliant drug for a perfect pocket, but if hitting that pocket does nothing to stop the protein's malignant behavior, the drug is a clinical failure.
So how does the AI handle that?
To solve this, the AI suite takes all the validated cavities and runs them through a multi-trait pocket ranking system. It classifies them as truly actionable based on 3 deeply integrated traits.
Okay, let's go through them. The first trait is ligandability.
Right.
This seems to be a pure physics check. It assesses the physical properties of the pocket using scoring functions, like D-score. So it's looking at the raw volume and the shape to ensure a drug can physically fit. But it also measures hydrophobicity. Why is hydrophobicity so critical for a drug pocket?
Well, hydrophobicity means water repelling, and most effective small molecule drugs are somewhat greasy or hydrophobic.
Okay.
If a pocket on a protein is highly hydrophobic, it naturally wants to expel water and bind to a greasy drug molecule instead. It creates this strong, energetically favorable magnetic pull for the therapeutic.
Oh, that makes perfect sense. It actively wants the drug.
Exactly.
The second trait it ranks for is functional relevance. The AI checks for crosstalk between this new hidden cryptic pocket and the known active site of the protein, which is called the orthosteric site.
Right. The active site is where the protein normally does its biological work, or in the case of a disease, its biological damage.
Right, the troublemaker site.
Exactly. So if we are plugging a drug into a completely different hidden area of the protein, the AI has to mathematically prove that the binding event will send a ripple effect through the protein structure, altering that active site.
There has to be a connection.
Yes.
Yeah.
There must be structural crosstalk. It has to incite a measurable biological effect.
And then we get to the third trait, which is evolutionary support. And I really have to push back here because this sounded crazy to me.
It sounds like a stretch, yeah.
We are talking about using supercomputers and artificial intelligence to design synthetic molecules to cure modern diseases today. Why is the AI suite looking at millions of years of evolutionary history to rank a pocket?
Because evolution is the ultimate stress test for biology. The AI analyzes the amino acid residues that make up this cryptic pocket, right? And it compares them across thousands of homologous targets, meaning similar proteins in other species, stretching back millions of years.
Wow.
It is looking for residues that are highly conserved.
Meaning they haven't mutated or changed over millions of generations.
Correct. And think about what that actually means for a second. If a specific structural rivet on a protein hasn't changed across millions of years of random genetic mutation, it means that rivet is absolutely indispensable for the protein's survival.
Oh, wow. If it mutates, the organism dies, or the virus dies.
Exactly.
Oh, I see where this is going. It's an evolutionary Achilles heel.
It is a trap the disease literally cannot escape. In traditional drug discovery, a massive problem is drug resistance. You hit a virus or a cancer cell with a drug, and it just mutates the shape of its binding site so the drug no longer fits.
Right, it adapts.
But if our AI designs a drug targeting a highly conserved cryptic pocket, the disease mechanism is completely cornered. It cannot mutate its way out of the drug's effect without simultaneously destroying its own biological machinery.
That is just a master stroke of engineering. We are deliberately targeting the load-bearing pillars of the disease's anatomy.
Exactly.
So, okay, we found the perfect, inescapable cryptic pocket. How does this fit into the broader reality of pharmaceutical superintelligence? Like, how do we take this digital map and actually manufacture a physical pill?
Right. So this is where the workflow hands the baton to the predictive and generative layers of the suite. Now that the pocket is mapped, a tool called ISO-DE steps in.
That's the Isomorphic Labs drug design engine.
Yes, ISO-DE predicts the binding affinity of potential molecules. It runs the heavy physics calculations to predict exactly how tightly a molecule will stick to the pocket. And the crazy thing is, it does this with a level of accuracy that exceeds the current gold standard physics models, like FEP++.
Wait, really? So it provides experimental-grade predictions without actually having to synthesize the chemical in a physical lab first.
Yes.
That saves so much time.
It does. And then it feeds those high-accuracy predictions as reward signals into a generative AI core, specifically a model called Proteína-Complexa.
Proteína-Complexa.
Right. This generative model was trained on a massive synthetic dataset called TID-Worm, which contains 3.5 million biophysical interaction clusters.
And for you listening, you can think of an interaction cluster as a highly specific 3D jigsaw puzzle piece.
That's a great analogy. So Proteína-Complexa uses this vast library of 3.5 million puzzle pieces to generate a fully atomistic binder, an entirely novel drug molecule designed from scratch to fit the geometry of that exact cryptic pocket.
It just builds it.
It just builds it. But the superintelligence suite doesn't stop at the digital design either. It then interfaces with an agentic system called CEPROAGENTS.
Okay, what do they do?
These agents act as virtual chemical engineers. They figure out the real-world manufacturing scale-up, they design the process flow diagrams, they calculate the reactor temperatures, and they determine the exact chemical synthesis steps required to physically manufacture the drug in a laboratory.
Unbelievable. And it even handles the legal bureaucracy too, right? Because regulatory paperwork is famously one of the most agonizing bottlenecks in drug development.
Oh, it's a nightmare. But the suite uses a tool called AutoIND to handle it.
AutoIND.
Yeah. It uses retrieval-augmented generation to draft the dense regulatory dossier required by the FDA.
And retrieval-augmented generation, or AG,
Yeah.
means the AI isn't just, wildly guessing or hallucinating the legal text.
Right, definitely not hallucinating. It actively retrieves data from thousands of past successful FDA submissions. It combines that historical precedent with the new drug-specific data to format airtight regulatory documents.
That's huge.
It is. It reduces the drafting time for these complex non-clinical summaries from around 100 hours of human labor down to just 3 or 4 hours.
From 100 hours to 3. This is where the sheer disruptive return on investment of pharmaceutical superintelligence just becomes undeniable.
Absolutely.
Because at the start of this deep dive, we talked about how the standard timeline just to get a drug candidate from target discovery to preclinical trials takes 3 to 4 years of painstaking human labor.
3 to 4 years, yeah.
But by tightly coupling these AI agents running all L sites to scan the proteome, Pocket Miner to triage the weak points, MSMD to prove the physics, and Proteína-Complexa to build a molecule, that entire timeline shrinks to between 12 and 18 months.
It fundamentally rewrites the economics of therapeutic discovery.
Right.
The bottleneck is no longer human intuition or trial and error chemistry. The bottleneck is simply the speed of computation.
Amazing. For those of you listening, let's just step back and look at the ground we've covered today. We started with an industry completely trapped by its own complexity, literally losing billions of dollars throwing chemical darts at invisible targets. And we've seen how a coordinated suite of AI agents can scan the entirety of human biology, identify hidden stress points, use advanced physics to flood virtual cavities with water and benzene, and then custom build a molecule that locks on to an evolutionary vulnerability the disease just can't survive without.
We have successfully automated the prompt-to-drug pipeline.
We really have.
And, as profoundly advanced as this multi-agent suite is today, we are rapidly approaching an even more radical threshold.
Ooh.
Yeah, we are entering the era of what are called hyperagents and Darwin Gödel machines, or DGMs.
Darwin Gödel Machine. That sounds intense.
It is. These are AI agents capable of metacognitive self-modification.
Wait, what does that mean?
It means the AI can look at the physics equations we gave it, realize they are inefficient, and literally rewrite its own source code to improve the very process of discovery.
Oh my God.
Right. So I really want you to ponder that for a moment. What happens to human medicine when the AI stops just finding hidden pockets, using our rules, and instead starts evolving entirely new laws of digital chemistry that human beings haven't even conceived of yet?
That is, wow. That is a wild thought to leave on. Thank you so much for joining us on this journey today. And as always, thank you for taking the deep dive.